Well, I think we have a complete committee. So today is the day for Jorge. He's going to defend his dissertation on modeling epidemics in structured populations. And I want to get this thing on the road as quickly as we can, because it is a lengthy presentation. We just crammed everything in there that we possibly could. The procedure today is like this. Jorge will be presenting. He can take questions if there are any during the presentations. And we are going to allow questions afterwards. Then I will ask those of you who are not on the committee to leave the room and leave the committee and Jorge to some one-on-one. And after this, we will ask Jorge to leave, and the committee will meet and make a decision. That's how we're going to run this show. And with this, I leave that to you. Go for it.
Well, thank you very much, everybody, for being here, especially committee members. I know summer just started today, so it's kind of busy. My committee members are Dr. Golden and Dr. Kara from biology, Dr. Benka and Dr. Thoreau, and my favorite professor, Dr. Miller from computer science. Today I will be defending my dissertation, which is titled Modeling Epidemics on Structured Populations, Effects of Behavior Changes and Immune Responsibility of Individuals. So, when I first joined Dr. Miller's lab, he was assigned, we were playing with the idea of what is the role of individuals in the spread of diseases? How the unique characteristics of individuals affect the spread of the disease. We first looked at the social behavior characteristics. From there, we started looking at how the physiological characteristics of the individuals can affect the outbreak dynamics. We were also interested in not only looking at the spreading in time, but also in space, to finally kind of put everything together and look at the effects of demographics in a region, in a geographic space. So the question we had is like, who are we really? So whenever we look at individuals, of course in the terms of epidemiology, we ask the question, how individuals are alike? And not only that, how are individuals the same? Can we say that they are the same based on their gender, based on their culture, their income? And specifically in the area that we're interested, we were asking questions like, how does the characteristic of an individual affect their interactions? Or how do they, why some people are sick for a longer period of time compared to others? Or how does the immune responsibility, the demographic characteristics determine the amount of time an individual is sick? We were asking questions as, how can we measure the propagation of the epidemic in a regional space, and how finally how the demographics will affect the epidemic progression? To make things a little bit more complicated, we were interested in interactions, because for a disease to spread, there needs to be interaction. So the question was, are all people interacting the same? And do we have the same chances to interact with each other? For example, or chances to interact with our wife, our husband, or kids are higher than actually, like this happy baby that have a chance to meet the president. So, looking at that, why was this important? Why we wanted to look at individuals, and especially involved in we know that epidemics are occurring seasonally, annually. We know that we have new threats like specific new diseases. We know that there are bio-terrorism, and it is required for the public health officials to develop new tools. We're proposing that computational epidemiology could create those tools. So, having a little background of epidemiology in general. Whenever we talk about an epidemic, we know that we divide the population into four groups. We talk about susceptibles, latent, infectious, and recovered. Susceptibles individuals are individuals that do not have the disease but potentially could get infected. Latent individuals are individuals that have the disease but have not developed it yet so they can infect others. We know that an infectious individual has the disease and is capable of infecting others, and the recovered individuals are individuals that have already either naturally or artificially caused the disease and are non-infectious or infected anymore. Additionally, we're interested in looking at the infectious periods. The infectious period is the amount of time an individual will remain in one of the different stages. The latent period is the amount of time the individual will remain latent. The infectious period is the amount of time an individual will be transmitting the disease to others. What really grabbed our attention was the fact that this is information from CDC, the fact that the values are not precise. We never have a 4 or a 5. We always have intervals. Now, we will investigate and talk about that in more detail later. The classic original epidemiological model, the SIR, and modification SLIR, includes 3 differential equations or 4 differential equations that represent different compartments. So what we're saying is, in this compartment we have the susceptibles, but not only that, we're seeing the changes in the susceptible population at every time step. So what this is saying with a negative symbol is saying that we're losing susceptibles by the interaction between infectious and susceptibles and a transmissibility. And we observe that in the infectious we're winning them. That's why we have them with a positive symbol. Now the problem with these models is that it's very difficult to include heterogeneous characteristics. We have found models that include them, but it's very difficult. So that's why we started proposing more computational approaches. The computational approach requires the understanding of a contact. As I mentioned earlier, if we don't have contact, then the disease cannot be transmitted. Now, it's important to define that a contact is disease dependent. If we talk about malaria, for example, we would say that the contact is a bite of a mosquito. If we talk about HIV, then we're talking about a sexual contact. So, and from there, we can also define the contact rate. The contact rate is the number of contacts the individual will initiate every time step. Well, the problem with this approach of having a standard contact rate for everybody is that in reality, people are different. Some people will, be more social or have positions in their job that makes them to interact with more people. And we have some other people that are more shy, so they will have less interactions. So the first parameter that we studied was the contact rate. But before we go to the analysis, the second, the first approach, we had a whole population in here. And what we did is we looked at all the possible interactions. So we said this is population, this is individual 0, individual 1, individual 2, individual 3, and so on. And we look at all the possible interactions between them. Now, the problem with this approach is we had a lot of contacts that didn't lead to anything. For example, if a susceptible and a susceptible interact, well, nothing is really going to happen in the terms of the disease. I mean, maybe they become really good friends, but there will not be anything related to the disease. So we improved this method in what we call the global stochastic contact model to only consider the contacts that involve an infectious individuals. Now, this type of contact will include also the ones that go from infectious to infectious and infectious to latent, just to maintain the consistency of the number of contacts or the contact rate. Interestingly enough, whenever we separate them, we were able to see that, for example, P3 likes to interact with P0 and P3 likes to interact with P4. And we were asking the question, why does P3 select P0 and P4? Why doesn't it select P1 or P2? Well, it's because there are some preferences. P3, maybe they go to the same church, or maybe they go to the same school, so that makes them to have a preference. How do we represent that preference? Well, we implemented something that we call an affinity matrix. The affinity matrix, to do the affinity matrix, first, we divided the population in groups. The groups are nothing else but an abstraction of regions, schools, different types of ages. So we divided our population in groups. We decided to group in 5 groups. Now, in these groups, in this matrix, what we see, we're saying the probability of A interacting with A is 0.19. So individuals from A interacting with individuals from A is 0.19. 0.39 from individuals A interacting with B, and so on. Now, we conducted some experiments to measure both the contact rate and the affinity. So what we did is we had a population, a small population of 1,500 individuals, and we divided in 5 groups. Now, we selected one group as the pivot group, and this group will be changing so we can see the variation of the effect of this parameter. I'll explain more in detail in this slide. So what we did is we have a general contact rate. So we said, for example, the contact rate is 200. So I have to distribute that 200 among my 5 groups. Whenever I gave 40 to the pivot cluster, the other ones have 42. Such that 40 times 4 is 160 plus 48 is 200. When I gave 180 to the pivot cluster, I gave 5 to the other cluster, such that 5 times 4 is 20, so 180 plus 20 is 200. The results are depicted in this figure. We observed that as in this figure we have the number infected by the pivot cluster group, the number infected by the rest of the groups in green, and the total number of infected individuals in red. What we observed is that the contact rate of the pivot cluster increases, it gets the chance to infect more people. Now, more interestingly, we found that whenever the contact rate is homogeneous, meaning it's 40, 40, everybody has 40, we have the greatest outbreak. What does that mean? If we consider that the contact rate, imagine we have the 5 boxes, and all of a sudden we still have infectious individuals in all of the boxes. But if we just give the contact rate to one box, we have all those infectious individuals that are not doing anything. So whenever we distribute it in all of the boxes, we have the highest infection. We did something similar with the affinity. We did the same. We have an affinity to the pivot cluster. So we observed we have similarity here. We have the number of infected individuals in the pivot cluster and the rest of the clusters and the total population. We observed that whenever more and more people are attracted to the pivot cluster, more people get infected. But interestingly enough, again, whenever we have a homogeneous affinity, we observe that the outbreak is at its maximum. And that is because, again, if we use the same example with the boxes, if we have susceptibles in all of the boxes, but we only are making everybody to look at one of the boxes, we have all of these other susceptibles that are not being attended, that we are not contacting. The third experiment we did is we look at the differences in the outbreak dynamics in a homogeneous affinity cluster. So in here we look at every group. So we have the 5 groups plotted together. We observe that the dynamics in every group are very similar. And the red line is just the addition of all of them. So whenever we increase the affinity towards one group, we saw that not only the pivot group, or the group that gets all the attention, has a higher outbreak, but also the outbreak in general decreases in size and magnitude, and increases a little bit in width. What is happening here is, again, since this group is attracting more contact than the others, it's just limiting the progression of the disease. In general, these experiments were telling us that we need to look at the contact rate and affinity. We are seeing changes in the outbreak dynamics. Now, afterwards, we started looking at the physiological differences. We were looking at the individual. Because so far we've been talking about groups of individuals, we're looking at populations. So we started to look at the individual itself. So whenever we started studying, we run into this graph. Well, something very similar. And we look at the viral load or bacterial load. We call it in the dissertation, you'll see viral bacterial load. We are not specifying the pathogen. Whenever the individual is healthy, we start seeing that the viral load starts increasing. We know that there is a threshold. Once a certain amount of pathogen is in the host, the individual will become infectious. Whenever the viral load will keep growing and growing and growing, but then there is something that it will make it go down. So what do you think is that force that is bringing this viral load down? Dr. Ivan, the system? Yes, If we imagine the immune system is just a force that is bringing this down. Now, thankfully, Dr. Bowen took me into his class for me to be able to understand all of these concepts. Because honestly, for us to be able to model or to understand the models of immunology, we need to understand the the concept, the biology. So I'm very thankful for taking this. Whenever we talk about the immune response, the immune system, we talk about two main components: the innate component and the adaptive component. The innate component is the one that is not looking for anything specific. It's always going to react the same no matter what pathogen it presents to. Now, the adaptive response, on the other hand, will have unique responses based on if it's HIV or if it's tuberculosis. Their reaction is different. So in this diagram here, we observe, for example, if there is a virus that enters into the system, there are certain cells and proteins that are patrolling the body, especially the macrophages, which are the first line of defense. They will phagocytize, which is they will digest the virus, then they will digest it and present it to other cells. In general, usually the T helper is the one that is presented to, or it could be presented to other cells. The T helper is like the director of the orchestra. It will be sending signals to the other cells so they can start mounting the defense, or regulating in case it's time to relax. The T cell will send signals, especially to the B cells. Again, it depends on the infection. That's why it's the adaptive system. And the B cells can produce secrete antibodies. And antibodies will help to stop the spreading of viruses because they will function like cushions, that they will stop the virus to attach to body cells. Now, the B cells also have other functions, but for us, this was sufficient to understand the models. Now, we also have a complement system, which is a protein that intensifies the function of the other elements of the immune system. And we have natural killer cells and T cytotoxic cells, which will create apoptosis and will kill infected cells, and then releasing the viruses. So once we understand those concepts, we were looking, okay, we go back to our graph, and we were, okay, what could bring that down? What makes it from the individual makes it unique so that graph goes down, or So we started doing some research, and we found that, for example, the age is one of the main factors that determine the quality of the immune response. Such that the individual ages, if T cell and B cell functionality is reduced, the thymus, which is the place where the T cells are produced, it shrinks in size. The B cell population and the antibody diversity decreases, and some other. Now, we also know that in nutrition, if we have individuals with malnutrition, obesity, or low vitamin intake, they will have problems with their complement system or with the phagocyte functionality. Gender, we have some of the hormones that by nature, the androgens are immunosuppressors, and the estrogens are B cell boosters. Finally, physical fitness, we have individuals that exercise in a moderate way, they will have a better immune system than individuals that do not exercise or exercise in excess. Well, as I mentioned earlier, whenever we were looking at this timeline, we saw that there was a variation. Now, looking at this figure here, we observe we are plotting 4 types. We're saying, we have an individual that gets to the threshold, stays on time, and then, so we will have some type of immune response there. Now we have one that maybe comes here, it stays for even a really short amount of time and then goes down. We have individuals that don't even reach the threshold, and we have individuals that probably stay there, either are succumb to the disease or maybe stay for a really long time. Now, we were interested in looking at a model, something that allows to replicate this. We found the Waters model. This model is similar to the SIR in the sense that it deals with differential equations, and we have 5 of them. Now we have the uninfected cells, infected cells, the viral load, the antibodies, and the cytotoxic lymphocytes, or the T cells, that are cytotoxic. This model was interesting enough for us in the sense, or useful enough for us in the sense that not only mimics the progression of the disease in the host, but also quantifies some of the responses, such as, for example, the antibody neutralization rate, or the antibody development, or antibody decay, meaning that we potentially could rank all of these characteristics of the immune response numerically. Now, unfortunately, the data is not available. The data to create this calculation. Now, we're actually in the dissertation. We are based on our results. We're suggesting the collection of this data. Well, we're encouraging the collection of this data. Now, with this model, we still are able to mimic different responses. And here we're seeing an example in the model plotted in R, in which I am plotting two of the responses. So what we're proposing is, we propose the concept of population immune response. What we're saying is that this concept will capture the collective immune response of the individual in a population represented as the superposition of the individual immune response. So this is a framework of population immune response. What we have in here is we have the population. Now we have information from them. We have a census. We have information. Now, if we're interested in a specific disease, let's say, in which the age is a big factor or the gender is a big factor, we can group them. So we can have them here. Now, based on that information, if we know that they have certain age and gender, then we can rank their immune systems. Now, using immune competence module, we can mimic their immune response. Now, we can use the model that I just showed you, or we're actually using probability distributions in this occasion. This model will return the latent period and the infectious period for all of them. You remember we had the plot and we had the threshold, so we can measure how much time they are passing the threshold. Then we can model the outbreak in that population, and then we can obtain the outbreak dynamics. So in general, we're saying that the outbreak dynamics are actually linked to the quality of the immune response at the individuals, which is given by the demographics. So we define 4 types of immune responses, which is the hyperimmune, standard, hypoimmune, and immunized immune response. In this plot, as I mentioned, the Waters model allows us to mimic these responses. This is an implementation in the Waters model. We see the 4 of them. The hypoimmune response is immunocompromised individuals, the ones that will have a longer infectious period. We have the standard immune response, which is our individuals that are the average of the population. We have a hyper, which are individuals that stay a little bit less than the average, and the immune, which are individuals that they don't even realize. They never become infectious because they either have artificial or natural immunization. Natural meaning they have a disease and then their body has memory cells, or they have vaccine. In our examples, in our experiments, we divided the population in these 4 groups. 3 of them are based on age. This information comes from the World Health Organization. So what we did is we have adult, children, and elderly. These are divided by ages. So 15 to 55 are adults, 0 to 14 are children, and 56 plus are elderly. Now, we are able to obtain their infectious period, the length of the infectious period for the influenza. And we can rank them, we can give them a quality of immune response. Now, by using probability distributions, we can mimic these values. For example, if we look at the adults, we can have a normal distribution that goes, a mean of 6 to have something that looks like that. If we look at the something that is similar to this, if we have a 7 to 8, we use a Levi distribution, such that the majority of the values are in here, but it has a long tail to be able to represent that variation here. By using those parameters, we conducted multiple experiments to measure the effects of PIR. The first experiment, we just considered all of the population to be part of one group. So we said total population are adults, the total population are children, the total population are elderly, the total population are immunized. So, and here we observe that not only on the number of infected individuals we see changes, but we also see changes in the length and in the severity of the outbreak. For example, we see that, and here for the adults we have only 637, but for the elderly we have almost the whole population. It was a population of 4,000. These are the results of average of 50 simulations. Now, in the second experiment, what we did is we uniformly distributed the population, so every group had a similar number of individuals. So in this population, we had adults, children, elderly, and immunized. And then these individuals could interact in a homogeneous way, meaning everybody, there were no preferences. So what we did is we looked at the infected members, and it was very interesting. Since we're dealing with homogeneous interactions, then pretty much all of them are very similar. So all of the groups are getting infected similarly. However, the elements that are infected by changes. So we observe that the individuals that have the longest infectious period are the ones that are infecting more people. And in here, we plotted this other series in a cumulative way. Well, considering the important role of that group that has the longest infectious period, we incorporated vaccination strategies. These vaccination strategies, we did a 10% such individuals that were from the elderly population, we immunized them. And then we did 20%. And interestingly enough, we found that the difference between doing 0 and 10 is not as big as doing 20. The difference between 10 and 20. Not only we found a difference in the number of people that the number infected by this group, but we also saw differences in the outbreak dynamics. We found that the epidemic was less severe, meaning that the magnitude was shorter, and it was a little bit longer. It had a longer tail, considering we had less individuals that were what we could call the aggressive group. So, in general, we found that PIR was also telling us something. We should look more into detail. Well, with the tool that we have, the experiments that I've been showing you, we have been looking at the length of the epidemic, we have been looking at the number of affected individuals, we have been looking at the day of peak, number of affected as peak. But all of this, we're looking only at temporal space. We were interested in what happens at the geographic space, what happens at the geographic context. We have found methods of people, simulating in a geographic context. However, we were interested in looking at how the disease moves in a geographic space. So, what we are calling the epidemic trajectory. When we look at the word trajectory, we think about movement. So moving from one space to another, a trajectory. So we started looking at how to define an epidemic trajectory. So we look at the different types of paths. Well, the first path is a single branch path, meaning an individual that is infectious goes and infects another one. It's like if I have the disease and then Joseph is there and I pass the disease to Joseph and then I immediately get well, but then Joseph has the disease and then he infects him and then Joseph gets well, which was not very realistic because in reality, if I have the disease, I potentially could infect everyone in this room. But hopefully, I don't have it. So we will be talking about a multi-branch path. Now, whenever we talk about contiguous versus non-contiguous, so if these lines would represent regions. So if we think about individuals being schools or houses or census blocks, we would say, what are the different cases? Contiguous would be, I only interact with people that are in my region and in my neighboring regions. Versus a non-contiguous in which I can go beyond my borders. Which is more realistic in the sense that people, can take a plane and, go to border places or, commute. So we're looking at non-contiguous process. Now, that non-contiguous process we have to represent somehow. We look at a fuzzy neighborhood. The fuzzy neighborhood is a network in which an individual from a region can reach other individuals from other regions based on a probability function. Now, this probability function, we look at three of them: the inverse distance, the inverse distance squared, and the normalized inverse distance. So that what these represent, they are telling us that if the size of the population, the bigger the populations are, the higher the chances of them to interact. But at the same time, the distance, the closer they are, the higher the chances, the shorter they are, the less the chances of them to interact. So, for example, in this example here, which Mario will show us, we look at, where here are you mentioned. So, from Denton to New York City, so if we look at cities, we will be talking probably, 1,500 miles. And from Denton to Nampa, Idaho, we're also talking about 1,500 miles. Now, the chances of people from Denton interacting with people from New York are higher just because in New York we have 8 million people versus Nampa, which has about 100,000. Now, if we do the same thing between Denton and Dallas, even though New York has a higher population, just because Dallas is closer, we will have higher chances to interact with people from Dallas. Well, so we defined an epidemic trajectory as a multi-branch path of a continuous process that spreads in a fuzzy neighborhood. And here we have that example, in which we see that it's multi-path, and we see that it's going beyond. There is a non-continuous process. Well, how to define the geographic space? How to represent the geographic space? Well, we look at two different methods. The first one, which the examples, the experiment that I will show you in this first part, we're using this approach. We use a grid. This grid represents school, allocation. In this location, we have numbers such that 26 individuals live here, 35 live here, 12 live here, 0 live here. When you have zeros, we probably talk about lake, some areas that nobody lives in. The second approach, when we talk about the geographic space, we use census blocks. Census block is the finest granularity of information we can obtain from the census. So, in this example, this is actually Dallas County. So, how did we do it? We developed a model in which the disease is transmitted at the regional level. So, in the previous model that I showed you, the individuals are the ones interacting. And here, the regions are the ones that interact. How do they do that? Well, every region, let's assume a region has 3 individuals. And the 3 individuals that are in there, 2 of them are infectious. Again, we only consider infectious contacts. So what we look at is at the contact rate. So for example, if individual 1 has a contact rate of 20 and individual 2 has a contact rate of 10, we're starting 30 contacts from that region. So what happens here is we count all of those interactions. So if I'm in region 1, in region A, and I interact with Mario, which is in region B, I would say that there was 1 transmission of the disease. Well, I interact and I actually transmit the disease. We're considering transmissions. Now, if Mario was in B, and I have 12 siblings, and Mario interacts with 12 of them and infects all of them, then from B, which is Mario, which is in B, will infect 12 people. Now, this is, at the end of the epidemic, we can create a table like this, which can be represented as a directed graph. In this graph, we are representing exactly the same thing. For example, B12, we're saying from B, there are 12 transmissions that are going to A. And similarly, from A, there are going 1. There's only 1 transmission that is going to B. From this graph, we can extract some information. We can extract the out degree. So we can say from region B, the out degree of this region is 30. And we're calling that the force of infection. So some regions, like, if it has a 30, it has a 30 force of infection of 20. So it has a greater or smaller force of infection. We're calling the in degree as the transmission trend, meaning how much of the disease I attracted. Now, from there, if we look at the whole population, because this would be the out degree of a region, we look at the whole population, we can obtain the force of infection of the whole trajectory, such that the force of infection for group A, and the force of infection from group B, and the force of infection from group C, and so on. And we have it in a set. Now, from this set, we're interested in its properties. So, to do that, we first conducted 500 experiments, 500 simulations, and we obtained their sets. We obtained their force of infections. We had 27,000 individuals, 300 in each region, and we had 100 regions. Now, we put 10 of them with 0 individuals, such that, again, relates. And we had homogeneous contact, so everybody had the same chances, and also there was, everybody could interact. You know, there was no interaction coefficient. And here I am depicting the density graph, so you know the probability of being in any of these points. For the 500 simulations, we observed that we had a really high 0. What happened here is that some of the simulations led to what we call no average. So you know, I start with average and infect one person, then another person, and then that's the end. So that's what we see in here. So we remove what we call the no average. And we have this segment. Now we're interested in the average dynamics, so we remove the no average. We look in here and we still have some zeros, but these zeros are actually the regions that have 0 individuals. So we remove those as well, and then we have the 500 out degrees, or the 500 forces of infection. Now, from here, we can look at the mean of this force of infection. So we can say that this mean is actually the mean of the population of the force of infection with a standard error. Now, once we have developed this method, we were interested in comparing the trajectories between multiple populations. So in this experiment, we were looking at the consistency of the method. So what we did is we had a population with uniform interaction coefficient, meaning everybody had the same chance to interact with everybody, and one with the interaction coefficient that one of the interaction coefficients that I presented, the one with the population at a distance. And then we looked at the mean force of infection. Now we were interested in making sure that the effects that we were looking at were not caused by randomness, that were actually caused by the effect that we were implementing. So we did 2 approaches. The first one was the Welch t-test, which is a modified t-test that determines if the sample have the same population mean. Now we use the Welch t-test just because the variances are not assumed to be homogeneous. Now we are also measuring the variances, so we wanted to be sure that it's not assumed from the very beginning. Now they also assume independence of the data. Now what we found is that if we run one simulation in here, it will not affect the outcome of this other simulation. So we consider the data to be independent. And more importantly, the variations to normality have little effects. Now our data sometimes is normal, sometimes it's not. So we wanted to make sure that we could still run a test that will return correct results. So this test consists of first determining a null hypothesis, which in this case is saying that the 2 samples have the same population mean. Then we calculate a t statistic and we compare the t distribution. Now there are multiple t distributions, and the one that we use is determined by the degrees of freedom, which is based on sample size. From there, we calculate the p-value and the confidence interval. Now, if the confidence interval it has the value of 0 in there, it's telling us that the null hypothesis is accepted, meaning that the 2 samples are the same. They come from the same population mean. Then, whenever we have a p-value of less than 0.05, since we're using a confidence of 95%, we will say that the null hypothesis is rejected. So, we also, additionally, we implemented the Levene test. The Levene test will determine if the variance of 2 samples come from populations with equal variances. The assumptions and why we selected Levene is because it's robust and it has low sensitivity to normality, meaning that we can use different distributions besides the normal distribution. And in the implementation we used, we conducted a t-test on modified data sets. So we have data point 1, and we subtracted the median. Data point 2, we subtracted the median, and we created a new data set. And on that new data set, we conducted a t-test. The results are here. And in the first experiment, we had a sample of 500 with uniform interaction coefficient, sample of 500 with uniform interaction coefficient, and then we compared them. And we found that they are the same. Again, since the value is greater than 0.05 and the value of 0 is in here, we said that they're the same. Now, the Levine test is also telling us that the two populations are the same. Now, that is a little bit, again, to test the validity of the method. I mean, we're pretty much comparing the same thing, but we still wanted to be sure. Now, this, whenever we incorporated the other parameter in which we have a sample with a uniform interaction coefficient and a sample with a normalized interaction coefficient, and then we compared them, then we found them to be defect. So this gave us confidence that this method was actually capturing those variations, and they were not caused by randomness. Finally, we were interested in looking at the progression in a region. And not only that, looking at the demographics and how they affect the characteristics of the outbreak. So to do that, I implemented a modified global stochastic contact framework. This was developed in the lab by Sarat and Dr. Nitter. This modified version allows to have a spatial, not all are the modifications, but this framework allows to spatially distribute the individuals based on the census data. So we know where the individuals are based on where they live in real life. We also can assign them demographic characteristics. We know their ages, we know their incomes, we know a lot of their information. Again, in this model, it's similar to the previous one in which interactions are between regions, not between individuals. What happens is if there's an infection from my region to the region where Maren lives, we will look at an individual. So the regions don't get infected, individuals get infected, but the contacts are between regions. This model also has a visual component that, for my purposes, I am not, for this dissertation, I'm not using. But it's important to know that it's there, given that the visual component shows how the disease is being transmitted to the different regions in a color code basis. You know, the darker the color it is, the more individuals are infected. Now, this potentially could be a tool to, you know, investigate other effects. The population is divided in by census blocks, again defined as granularity. And in every census block, we have groups. We use the same groups that from the other experiment, in which we have adults, children, and elderly. Now, I have to be honest. In the proposal meeting, Dr. Goldman gave me a very interesting suggestion. And he said, why don't we test PIR in two distinct regions, in two regions that have completely different demographics? Well, took me to Florida. Sumter, Florida has the highest elderly population in the country, with 63% of the population are above 56 years. Now, we also see that the children from 0 to 14 are 7.4, so it's mostly elderly. Now, we have almost 100,000 individuals. Now, I compare this with Denton, in which we have most of the population is adult. Now, not only the demographics are different, but also the distribution of the population. We found something very interesting in Sumter. In Sumter, we have almost, 1,400 square kilometers of area. Now, if we look at this little square, which has the size of 69 square kilometers, we have 50% of the population lives here. Not only that, out of that population, we have almost all of it are elderly. The rest of the 50% of the population live here, and we have more variation. On the other hand, when we look at Denton County, if we look at the entire population, this is color-coded. The darker the color, the more individuals from that group. We see a more uniform distribution. We see that, there is not a definitive place where the individuals live. Of course, we see a high density of the adult population just because there are more adults, but they are not located in a specific location. So, we were interested in examining the effects of PIR. What did we do? We had experiments with PIR and experiments without PIR. So I would call those without PIR with uniform immune response. So all of them are adults. Versus the one in which PIR, we assign their quality of the immune response based on their group membership. We did experiments in Sumter and experiments in Denton. And then we look at the variations in their trajectories. So, again, we tested the method by looking at uniform immune response. So, experiments with uniform response, we compared them, and we found to be equal. We did the same thing in Sumter, we did the same thing in Denton. Then, we did experiments with population immune response, and then we compared them, and we found again, that they're equal. Well, the next step was to do experiments with uniform immune response and experiments with population immune response and compare them. What we found is quite interesting. We found that the t-test is telling us there is a difference. So it's telling us that the mean shifted. However, it's telling us that the shape of the distribution remained the same. So we had, it was looking like a normal distribution. Well, the next step was, we look at, yes, this is happening in Sumter, yes, this is happening in Denton. But again, going back to the situation from above, we want to be able to compare both. We just don't want to compare, what's happening in Denton. We want to compare both. So what did we do? First, what did we do? First, we looked at the disease dynamics. So we saw that there was a great change in the disease dynamics between epidemic dynamics between a homogeneous and between the one that has population response. Now, it's important to know that this is 10 times smaller than this. The interesting aspect here is that we don't only have a greater outbreak, but we have a long tail. What is happening here is, since we have a longer infectious period, we're actually reaching regions that otherwise we would not be able to reach with a short period, a short infectious period. Well, whenever we compare, we wanted to compare the mean funds of infection. Remember when we had all those distributions and we look at the mean, we wanted to compare the means. So whenever we had a homogeneous in Sumter, the mean force of infection was 41 and in Denton was 50, PIR was 66 and 85. However, we cannot compare them just like that because Denton and Sumter are, we said they are different. So we have to normalize them. How do we normalize? Well, what we look at is what is driving this force of infection? Well, the population size and the infectious period of the individuals. Well, since we have different groups, we have to calculate this denominator by looking at the population of adults times the infectious periods of adults, plus the population of children times the infectious period of children, plus the population of elderly times the infectious period of elderly. That way, we normalize our values, and that is what we're presenting here. Now that we have apples and apples, now we can compare them. What we found here is very interesting because we found that the variation in Sumter is actually 6 times greater than the variation in Denton, which is telling us that the effects were felt greater based on the demographic distribution and the population distribution that exists in Sumter. Well, as a summary, the contributions from this dissertation are, whenever we look at social behavioral characteristics, we found that including heterogeneous characteristics and diverse behavior in an average simulation are crucial to achieve a more realistic and complete prediction of an epidemic. Whenever we look at the physiological differences, we highlighted the augmented role of the immune response as a function of the social demographic characteristics of the population and their role during the epidemics. When we look at epidemics in time and space, we define epidemic trajectory and we presented a method to statistically compare variations in the properties of two trajectories. Finally, in the modeling outbreaks in geographic regions, we highlighted the importance of incorporating demographics and consequently immune responses or immunological characteristics when investigating the spreading of an epidemic. In general, we are recommending, we're suggesting the public health officials to carefully examine the effects of demographics and corresponding physiological characteristics when preparing for an infectious disease epidemic. And future work, this work it had open questions, such as study the clustering of regions. We know, for example, region A will have, Denton will have probably more interactions with Denton, and it will be constant. How can we take advantage of that knowledge in the methods of representing? How can we link those clustering of regions? Number two, transmission trend. Everything I showed was on the out degree, the force of infection. What about the in degree? The in degree is ruled by other characteristics, such as the affinity. So it would be interesting to observe the effects that happen on the transmission trend. Estimating the quality of the immune response. I presented two methods, the mathematical model and the probability distributions. Now, as I mentioned earlier, we need the data because we found that these methods are important and collecting that data is crucial for us to have a more realistic estimation. Finally, the methods that are presented are looking for a specific parameters. We're looking at PIR, we're looking at the affinity, we're looking at the contact rate. What happens if we start combining? What happens if we look at the effects of multiple of these parameters? And with that, that's all I have. Thank you again for your time. And do not hesitate to ask if you have any questions.
Nice timing.
Oh, that's it?
Worked well. Are we having any questions?
Yes.
That was a good presentation with the mind and the science and AP was a good combination of presentation. But I have one question about taking your concept, the model you're presenting, and thinking about the current outbreak that we are experiencing in Saudi Arabia, MERS, coronavirus. So what can you tell us about how your model can be applied to that kind of, especially the population is very homogeneous, right? And very restricted in mobility within Saudi Arabia. So looking at the context, looking at the population demography, how do you think that model can help us?
I think it can help us in the sense that we can learn the way on how we spread there compared to how we spread in other regions. You know, the sense of the demographics of Saudi Arabia are going to be unique in the region. So wherever they hit the region where it hits, what we're saying here is, you know, if we have an influenza outbreak, again, this is not real data, we will see differences. We will see different magnitudes of the epidemic. So I think the same approach could be implemented there. Now, the SARS, the MERS that you're mentioning is not as easily spread as the influenza. However, those characteristics that are unique to the individuals, maybe those are the ones that are telling us who is actually susceptible. And that's actually another factor that we could extend our work into looking not only how much time you're infectious, but also how susceptible you are to the disease, which is something that we could expand to look at that kind of infection that we're referring to. Yes?
Yes, so what would be the main computational technique that you feel contributed to this work? And then sort of as a follow-up question, what was the main thing that you had to solve from a computational problem to do this?
The main, I think, I would say in 6 years, the main computation aspect was modeling. It was the learning how to model and what is to model. Now, whenever we were running the experiments, I actually was running, I didn't present the results, but Whenever I talk about infectious contact on the whole population and infectious contact only with infectious individuals, sorry, contacts between the whole population and contacts in the population only that is infectious, we start looking at how to reduce computation. So we were looking at our models and how to make them optimal. Whenever I run the last experiment, when I look at the internal center, originally I was looking at one run for every 3 hours, which was a little bit too much. After experimenting and doing modifications to our algorithms, we were able to do it in 20 minutes. So I think a lot of the contributions to computer science are more on the modeling side of things. And I would remember if I asked those both questions.
Well, I think that was almost addressing both. Maybe I just asked a follow-up. So let me just try to summarize. My first question was really what was the main computational technique? You mentioned modeling. And what were the main problems we had to solve? You mentioned the optimization.
Yeah.
So would you say that, I mean, if we were just to sort of categorize this broadly in terms of algorithm work versus, let's just say, high-performance computing or something like that, where would the main problems have been if we look at those kind of categories?
Yeah, we would be more in the algorithm aspect. Now, high-performance computing, these models are always, they can always be modified to do high-performance computing. Now, for the purpose of a given time, I was not able to explore that aspect. Some of the members of the lab are actually exploring that, but I was not. The only thing I did is I actually, used clusters, and I was able to use different processors and, cores and stuff. But I didn't get to explore that. So I stayed in the modeling aspect and in the algorithm development.
But in some sense, I think data representation was the question was how do you capture and represent a new continuous trajectory? That I think was a big
Yes.
So that's really more on the representation side.
What kind of user interface does your software have? Is it convenient or learning parameters, conducting experiments?
So I have every chapter has a different model. Most of the models are non-user friendly. They're mostly text-based, except for the last one. The last one is user friendly. It has a user interface. And as I mentioned, it has that visual component, which I am not using, but it's just commented out. You potentially could look at the outbreak. So I was more interested in the analysis of the data, but potentially all of this can be, expanded and have a user interface. As I said, the last one has a user interface.
Yeah, maybe a follow-up on Brian's question. So on the modeling aspect, I appreciate that and observe that. So you basically set more of the parameters to try to understand the behavior of these models. But from a biology point of view, biologists don't have the. So I want you to understand, don't have this unrealistic expectation of biologists for them to measure those parameters you're talking about. For example, the immune system, how the immune activities. That's tough. So for them to get accurate and measurement, sometimes it's very unrealistic. It's not a matter they don't want it, they don't know how to do it, but they're calculating. So my question is that how do you think about using the data you observed? Like think about you already observed how for some disease you don't know the infection rates, how widespread they are. How do we actually turn around to estimate those characters in the models rather than setting the models? So right now the model you're trying to show that those characters matters. So that's what you're trying to show. But I think that one step further you want to ask is how do you actually, based on the data you have, try to estimate the pyramid itself?
Yeah, that is true. First of all, finding the data is very difficult, and second, transforming it is complicated as well.
Yeah, assuming you do have
assuming you have the data. Now we have found papers in which they use different methods to run the immune response. We actually attended a talk in which they were measuring the immune response based on what people were writing. But they wrote documents and they read of them, and then they did measures of different presence of antibodies. So we have found that there are different methods. And actually, in the literature out there, whenever I was doing this research, we found some parameters. We found some parameters, but not enough to really rank as much as we are desiring. As much as saying, you know, we have found, for example, some of the antibody quantity. You know, those can be measured. But at the same time, there are ranges. You know, some people, whatever is considered normal for one person or a certain amount is different for another person. So, of course, this would require, you know, a deeper study. But what we're saying is that that linkage exists. You know, there's a link between those demographic characteristics and the quality. Now, as I mentioned, we have observed, we have looked at some methods. And we haven't seen a complete method that will look at all of them. But what we're suggesting is that we potentially could look at them. To look at, we're highlighting the presence of the linkage between those two and how they affect the algorithm dynamics, meaning that we really need to go and look in more detail.
Now, your question regarding the estimation, that's in some sense exactly what we did through distributions. First, we got, of course, the time intervals that were recorded, and we fitted the distribution on those, and then we drew from the distribution. So, in some sense, that is the estimation that you are relating to.
So you're saying you chose the parameters in your statistical model based on fitting data?
I have a question. Your model was basically graph-based, and you were looking at moving these out and these and like very much on the model of the phenomena. It is not unusual that people do similar things in these graphs, let's say in the social networks or natural language processing or Google itself is the web, to use different centrality measures that are a little bit more focused on, like region, for instance, on how influence propagates rather than just counting the number of outings would also count from who they come and how influential they are. Do you think that some future work would be interesting along those lines for spreading the epidemics?
Totally. I think whenever we open the can of worms with this idea of the epidemic trajectory, actually it looks like another student from the curriculum will continue this work. So I will be involved as well, because it seems we were able to reflect that trajectory as a graph. And as we're saying, there's many. What we need to always not lose track is what does it mean? You know, the algorithm means the course of infection and the course of infection. So as long as we don't lose track of what it means, it's not a graph anymore. But the algorithms for the graph, we can still use them. Because, for example, the clustering that I referred to in the future work can be approached with those kind of algorithms. But as long as we don't lose track of that is the force of infection, that is the transmission trend, and then yes, we truly believe that we can expand this work with those kind of algorithms.
Paul, to fill in a great point, however, keep in mind that the out degree, specifically the out degree, is a measure of how many successful transmission a region can generate, which is a function of how long the individuals in that regions are infectious. So it measures directly the effect of an extended infectious period due to diminished immune response. So it is actually a straight measure.
Right, but if you use some of the stronger centrality measures, you might see hidden parameters that you didn't think about.
That's right. So that may be correct. But for the measure, in terms of the infectious period, that is actually the measure.
But for in general, without putting PIR, then yes, if we just look at PIR, then it's a different story.
Anything else? Then in this case, I will be closing the open part of this defense, and I will ask those of you not in the committee and the ones who have joined us online to leave the room. Martin, I think you have to turn them off.
Yes, so they leave the room. Oh my God.